Hello and welcome to another episode of TWiML Talk, the podcast where I interview interesting people doing interesting things in machine learning and artificial intelligence. I'm your host, Sam Charrington. If you're listening to this podcast, you probably have an opinion about AI and where it's headed. I personally think AI will make a huge difference in how we approach both our work and personal lives. But as of today, the biggest impacts for me are in the little things. But enough of what I think. I want to invite you to join the conversation. Jump on over to twimlai.com/myai to let us know what you think about where personal and home AI is headed. Sharing your thoughts takes just 2 minutes and qualifies you to win some great prizes. In this episode, I'm joined by Charles Onu, PhD student at McGill University in Montreal and founder of Ubenwa, a startup tackling the problem of infant mortality due to asphyxia. Using SVMs and other techniques from the field of automatic speech recognition, Charles and his team have built a model that detects asphyxia based on the audible noises a child makes upon birth. We go into the process he uses to collect his training data, including the specific methods they use to record samples, and how their samples will be used to maximize accuracy in the field. We also take a deep dive into some of the challenges of building and deploying Ubenwa's platform and mobile application. This is a really interesting use case, which I'm sure you'll enjoy. Let's go. All right, everyone, I am on the line with Charles Onu. Charles is a PhD student at McGill University. Charles, welcome to This Week in Machine Learning and AI.
Thank you very much, Sam. I'm happy to be here.
It's great to have you on the show. I had a chance to see your presentation on, I think the project was called Ubenwa at the NIPS Black in AI workshop, and it was very well done.
Thank you.
And I'm sure you'll tell us a lot more about that project. But before we do that, why don't you tell us how you got involved and interested in machine learning?
Well, that story is very intertwined with the project Ubenwa that you just spoke about.
All the better.
Yes, yeah, because indeed it was in my bid to pursue that project that I got into machine learning and took side road from my original path.
Okay.
So I had my undergrad education in electrical and computer engineering, and when I finished my undergrad, I worked as a software engineer. It was during that time that I was volunteering with an NGO called Enactus, and then eventually co-founded one called Fisher Foundation with some colleagues in Nigeria. So I studied in Nigeria. And that was when during our work in the local communities, we had projects across agriculture, healthcare, and a number of other domains. And we came across the huge challenge of birth asphyxia, which is essentially when the baby does not breathe well right after birth. Mm-hmm. And it was such a huge cause of infant mortality in our communities but also in many parts of the world, and particularly in developing regions or resource-poor settings. So it was, it was in a bid to see what causes this problem, how could we address it. I came across research that, first of all, detailed that one of the big reasons why asphyxia is causing such huge casualties in developing countries. So about 1 million babies die every year from asphyxia, and another 1 million suffer severe lifelong disabilities like brain damage, intellectual disability, cerebral palsy, and so on. Mm. And one of the reasons why the casualty was so high was because in most resource-poor settings, the means for clinical diagnosis was too expensive, too resource-intensive, and was just not happening. Yeah, and so there was a need for something, a low-cost way of being able to screen babies early enough for whether or not they are breathing well. Because when it's severe, it's easy to know. The baby usually would not cry when it's not breathing at birth. But in many other cases, it's not as severe to cause the baby not to cry, but it's there and it's causing damage to the brain through oxygen starvation. And so the work we began to develop was based on clinical research done by doctors in the 70s and 80s, way back. And, they had looked at cries of infants suffering several pathologies, and in particular asphyxia. They used spectrograms back in the days. And they were able to notice that several characteristics of the cries, the fundamental frequencies and melodies, and very important frequency components of the cries were different, or in general, in the population of babies that had some of these pathologies from those that, were okay, were normal. And in particular, in the case of asphyxia, it's even more pronounced because breathing and speech are coordinated by the same region of the brain. And within our vocal tract as well, the same set of organs are oscillating when a person is breathing and speaking. And so the connection is so intertwined that if the baby is not breathing well, there is a manifestation in the frequency patterns in his cry. And so it was upon, I'm giving you the long-winded story of how I came into machine learning.
No, this is great.
It was upon discovery of this that I first of all heard about pattern recognition as a discipline.
Okay.
And I began to study and take courses myself to learn more about it, and slowly began to start what has become Ubenwa as a project now. Yeah.
Oh, wow. And you recently published a paper on this that won a Best Paper Award in the Machine Learning in Healthcare workshop at NIPS. Is that right?
Yes, that's right. Yes, our paper won the Best Paper Award at NIPS in 2015 NIPS, actually. And at the last NIPS, we published a new one, which was a follow-up to that paper, in which we had moved from algorithm into an actual deployable device in the form of a smartphone application. Yeah.
Okay. So maybe we can dive into that project, and you can tell us a little bit about in a little bit more detail how you approached it. You mentioned that the research came out of the 70s and 80s. Is this the research that identified these fundamental relationships, or were you somehow able to borrow data from that research as well?
Yeah, well, this research, first of all, told us that there was something to be explored there, because at the beginning, the last thing I would have thought of was to consider the infant cry as a useful signal for a diagnostic tool. And so it was really serendipitous, I'd say, to have stumbled upon one of these papers and to see that there was indeed a string of studies between the 70s and the 80s exploring the use of spectrograms to analyze the infant cries. And there was this winter afterwards in which nobody seemed to care again about what the infant cry could be used for. And my assumption was that at the time there were no concrete methods for transferring this knowledge or this hypothesis into useful applications as we now have today, with machine learning being so well developed as a field now, and several low-cost technologies like mobile phones that was not conceivable at the time of the 70s. So my thought was that was why some of those studies did not scale forward.
When you first came across this paper and got the idea that this might be applicable to this problem, you mentioned that you started taking a bunch of courses, but what was some of the first concrete things that you did to try to apply this knowledge that you came across? Yeah,
well, one of the first things I did was, I had a doctor friend, and we had long conversations about it. I tried to understand physiology of the infant breathing system from him, and first of all validate if this was something worth pursuing. And we together, worked on the early part of the work that we eventually did. Mm-hmm. But the next challenge too was, how do we find data to validate this hypothesis that the infant cry holds information about the presence or not of asphyxia?
Right.
As you know, machine learning is based on the ability to use data and learn from it and be able to generalize from that going forward. But as you probably know as well, in the medical space, acquiring data can be an extremely resource-intensive process, both in terms of time to get approval to interact with actual patients, or time it takes or the amount of cost or funds it would require to conduct the whole the study to acquire this data. And so one of the first things we sought was to find if someone had, by some chance, been collecting data of infants in the world. At the time, we were only interested in, say, if we only thought we'd find, cries of babies that were normal. And if we're able to find that, at least, we thought that would be a good point to begin to start modeling the inherent characteristics of cry, and to begin to phrase this as maybe an anomaly detection problem, where we have a good model of what a good cry should be, and we can say that any new sample that doesn't fit this model is likely an anomaly that has to be checked. That has to go for further verification. And I'm referring to babies here, and that could be a useful screening tool that first responders could use to transfer babies for tertiary care.
Right.
So we searched, and eventually we came across the database that we use for our work. It's called the Baby Shilanto database, and it's collected across a set of specialist hospitals in Mexico. And this team of doctors too, who had been tracking several pathologies in babies, and they had collected the cries of babies in several states, from normal babies and in different states, like hungry states, just a resting state, when receiving some measure of pain through maybe blood sampling. And also they collected pathological states as well, asphyxia, deafness, and a number of other conditions.
Okay.
And so we reached out to them, told them about the work we were trying to do, and they gave us access to the subset of their data that had the normal baby's cries and the asphyxiated baby's cries. Yeah, it was a fairly small data set, about 69 babies in total, but over 1,000 or so recordings. And that was what we used to develop our first work that really showed that there was promising signal in the cry that we could use to develop possibly a tool for diagnosis. Yeah.
How did you use that data to develop a model? What kind of model did you end up pursuing, and what was your general approach there?
Yeah, that's an interesting one. So one of the primary subdomain of machine learning that we looked to was that of automatic speech recognition. As you can imagine, this is a very similar problem as well. Mm-hmm. Whereas ASR is trying to take a speech and take a person's speech and understand what the person is saying. In this case, we're not trying to understand what the baby is saying, but right. What the baby's body is communicating to us. Mm-hmm. And so you can think that the methods developed there would be useful. And so that was what we did. We adopted feature extraction methods, like the use of male frequency septal coefficients, MFCCs. So MFCCs are an extraction of key frequency components in a speech signal, and they've been used a lot in ASR because it uses the methods of discrete Fourier transforms to extract these key components that really puts aside the things that are not as relevant, the artifacts of speech that are not as relevant, mm-hmm. and brings into the fore the most important components of it.
Okay.
And so MFCs have been used a lot in ASR, and we use that for our speech recognition, feature extraction path. And we use support vector machines at the classification phase and combine this with nonlinear kernels like the Gaussian kernels and so on. So that made the core of our system in general.
So you had these 1,000 recordings. You passed them through some pre-processing steps that basically broke them down into frequency components, and those became your features for your SVM.
Yes, exactly. Yeah.
And how, beyond that initial kind of model, like, did you run into any challenges in doing that?
So any challenges in building the classifier, you mean?
In building the classifier, yes.
Yeah, well, the good thing was, we've had this data that had been collected. It had been, someone else had gone through all the work of getting clinical approval for it to conduct this study, collected the data, filtered the data, they had annotated it by labels, which ones are the pathological samples and healthy samples and so on. So as you probably know, machine learning, there's a lot of work that goes into that early part, which we were fortunately saved from engaging in, at least at that time. So we spend most of the time, developing the classifiers and tuning and searching the space of what's the best solution within this range of parameters between the MFCC's extraction and the SVMs, hyperparameters. But ultimately we are now somewhat back to that first stage, because we're able to use that data to show the promise of this approach, to show the feasibility of this method. But now we need to, one, validate it, with data we collect ourselves. Mm-hmm. We need to get a larger data set to improve the performance. So the performance of the system we have now is about 90% on specificity and sensitivity measures. That's in accuracy in detecting the babies, the asphyxiated babies, and that's sensitivity. And accuracy in detecting normal babies is our specificity. Mm-hmm. And so, the goal is to try to one, robustly validate these results we have using samples acquired from a different geographical location, different population, and hopefully acquire this more data to use it to improve the performance of the algorithm, and begin to develop into our software very practical issues that would have to face in the real world, like making sure it's robust to noise. And because these samples were recorded in very controlled environment without background noise, mm-hmm. and just several optimizations by, for instance, in the presence of overlapping signals, several babies crying at once. Can we separate them? Define one. Can we optimize for the length of the audio signal that we require to make a valid diagnosis, and so on. There's a number of such things we're looking at now.
Okay. And now one of the tools that'll help you do all this is you were able to take the work that you did initially and then build a mobile app around this. Mel, tell us a little bit about that process and kind of what stage you're in with deploying this mobile app.
Yeah. So in terms of the development of the mobile app, one of the challenges is that a lot of machine learning, as you know, happens on Python and MATLAB. Mm-hmm. But those are not the languages that are used in mobile development in general. and so, but they really help with experimentation and fast turnover of the scientific process.
Right.
But thinking of deployment, we had to start thinking about, first of all, we had to decide upon what was the best mobile platform for a place like Nigeria, where I lived when we started this project. And Nigeria, Android phones are 90% or 95% of what people use. So that was our go-to platform to start with. Android, at the time, at least, was 2014 or thereabout. The main platform for development, or the main language for Android development, was Java. Now there's been C# portals to it, and I think Python possibly now. Yeah, but so one of the big challenges too was transferring our code to Java and maintaining efficiency, maintaining performance as well, accuracy, and just all of that thing. So it took quite a while. That was quite a bit of a few weeks or maybe months we spent on just that part of the work. But we're able to do it ultimately. I and my colleague Onesecond, who is also a software engineer, we're able to completely move our code and maintain performance, both in terms of time to diagnosis. We're able to maintain the original classification performance on the data set as well when we put it in the mobile application. So that was one challenge we had to face, and it was good. And we also, as you mentioned, that we faced this challenge too because we wanted to put the classification model on the device. We did not want it to require internet to do classification, because if we are trying to deploy in some of the poorest parts of the world, in Nigeria, that's also not so poor. Internet access is still very, very, very scanty in many places. And so if the device required internet to make a diagnosis, then purpose is, halfway defeated already. And we could go there in the long run, but in the immediate term, it was the most practical thing for our target groups. And so we went through that, and we got it onto the mobile app. So where we are now is a question as well in terms of validating that mobile application, and that's really a lot of what's taking it's what takes most of our time presently. So in Montreal, at the McGill University Health Center, at the Children's Hospital here, we are doing the validation exercise, a 1 year exercise with the doctors here. And the goal is to acquire more samples of babies who are experiencing different levels of asphyxia, from mild to moderate to severe, acquire control samples of normal babies, and then to use Ubenwa to validate against, to classify these samples essentially. So these samples are going to be the cry samples of these babies are going to be evaluated via the clinical methods of diagnosis. So their blood samples will be taken to be analyzed with a blood gas analyzer, and a doctor, a specialist doctor, would clinically determine, what to label these babies as, and then we're going to validate this against Ubenwa, and hopefully further develop the algorithm in that process. And the next stage after that would be to then take this to Nigeria and do field trials there. Our original plan was to do this stage in Nigeria. And it's still not a shut down plan completely, but we've faced many challenges with that. Just because, whereas the whole clinical process I just explained of blood gas analysis and so to confirm the presence of asphyxia, whereas it's a routine procedure in Montreal, in Canada. In Nigeria, it doesn't happen, which is the root of this problem in the first place.
Right.
Equipment do not exist in any of the public hospitals there. There's no power. There's no electricity for the most part to even use it if it was there. There's a whole workflow and process requirement that should be in place, or that we would have to put in place if we were to try to do it there. And all of this would cost both time, money, effort, and it would be a huge deviation from the ultimate, the precise focus of the project. And so after much forth and backs, we decided that we'll do the first stage here and then move to do field trials in Nigeria with the doctors we work with there as well.
And now it sounds like the samples you're collecting at McGill there's a high level of care, clinically in evaluating the samples. Are you also collecting them, with more professional kinds of equipment, or are you trying to collect the samples via the mobile app so that you can kind of match the conditions that you'll be collecting them within the field?
Yeah, that was indeed a decision point for us, whether to use the mobile apps, the mobile phones themselves to acquire the data, or to use specialized audio recorders. And ultimately, we went for special audio recorders just because we wanted to maintain, we did. We really want to maintain the recording quality across the subjects, across the samples. We want to be able to track things like the sampling rates and just exclude some of the immediate miscellaneous functionalities that come with a mobile phone. Also, we've had some slight issues with the ethics people. I don't know if this probably lands upon things I should not talk about as well. But yeah, there's also some ethics issues with using the phone right away for the acquisition, and so we're going to be using digital audio recorders to pick the signals at this point.
Okay, yeah, I'll take your word on it if it's something you can't talk about, but it's you certainly piqued my curiosity. I mean, I can imagine in the U.S. we have laws like HIPAA that require a certain level of standard of care with patient information. I imagine there's that kind of thing going on.
Yeah, things on that line.
Okay. Yeah. I mean, it can get complicated. You've got these samples that you're capturing with specialized audio equipment. How then do you address the issues that you mentioned previously? Kind of the transferability of those samples to the samples that are collected, via your mobile device, so your background noises, your overlapping. Like, are you you recreating these situations after the fact, or is that just reserved for a later stage of developing your model?
Yeah. At the current stage, we are going to be working on those optimizations more in a research setting. So we're going to simulate what noise would look like in a sense. We could go out, the sample. One way which we could do the case of the noise is to record actual noise signals in the environment we think the app will be used, and take that and use that noise to. I wanted to say noisy our samples further, but that'll be too much use of noise. But we'll use that to corrupt our samples essentially, and then work more from the research. So our research team is going to be working more on the research side of how can we improve the algorithm, make it more robust to noise. And I guess a more practical test of whatever we develop would happen when we are able to go through this phase of validation and begin to test on the fields in Nigeria. So we can put it down on the field and test it in reality.
How many samples are you expecting to collect in the current phase?
Oh, you shouldn't have asked that. Now I'm going to. I'm going to disappoint you if you've heard about machine learning data. But we are collecting a total of 100 samples.
Oh wow! Okay. It's interesting because I think perhaps the precursor to that question was, thinking about, whether this is something you would also evaluate like a deep learning type of approach. And I figured maybe one of the limitations would be the number of available samples and needing a much more efficient model.
Yes, that's correct. First of all, getting medical data is really hard. I work on another study at McGill, in which we are also collecting cardiorespiratory signals of newborns. And over a period of since 2014, it's taken us from then to 2017, December, to collect a total of 230 newborn data. So we're tracking, respiratory difficulties in the intensive care unit. And so the bottom line is, it's really hard. It takes time to get quality medical data, but that's also part of what turns us more towards classical machine learning and less away from automatic representation methods like deep learning, so that we can really have control over what features we extract and really make direct connections between the features and the cause they have on the system and their predictability as well.
On that note, are there, you started with this understanding from the prior research that the frequency components would have play a big role in identifying the asphyxia samples. In that kind of feature engineering process, feature identification process, did you identify any new features beyond what you found in the existing research? And in particular, was there anything that you found that surprised you?
I would say, because there's such a wide range of audio characteristics that was explored back then. But one of the features that really stood out the most was the fundamental frequency of the cry. So the fundamental frequency was one of the most studied elements, because it's like the very first thing that describes an audio signal. Even in music too, the fundamental frequency determines the key of a song, and so it more or less determines at what pace or at what pitch is this child crying at. And that I would say has been the most significant features consistently across our work and across the previous work that was done as well. Yeah.
Okay, so it's not there's not some kind of mysterious thing that's happening, like some, it's like just the basic, most fundamental thing that you would get out of this audio signal.
Yeah, but the interesting question comes in how it varies in the time across time. So the time varying nature of this signal.
Oh,
and
really?
That's why it's not a simple problem that you could set a threshold upon and say, okay, every value above this.
Right.
So that's really why machine learning was necessitated, because it's the complex time-varying nature of the signals and of these characteristics that it's just impossible to put down a rule that says, a baby that has this, and this is going to be normal and otherwise for asphyxiated.
And now how do you capture that time-varying nature with the SVM?
Yeah, so SVMs inherently are not time series classifiers.
Mm-hmm.
And so we've used a combination of several methods. So one of the things we did was use Gaussian kernels for the SVMs, which primarily deal with scalar features. But we then segmented the samples into several time chunks and used the instances in time as if they were independent for training. But at test time, we test the instances again independently, but combine their results to determine whether or not the performance has worked. The performance of the system is doing well at the level of the subject now, not at the level of the independent time instances. And so that way, by guiding the search basically through our parameters, by guiding the search with the ultimate performance on the subject, we can go to a better hyperparameter space that really optimizes performance at the level of the subject based on the nature of each of these instances in time.
Yeah, can you give me an example maybe of how that works? Like how does the relationship between these samples give you, allow the algorithm to key in on this time sensitive variation?
Yes, so you could look at it as imagine we recorded hypothetically the length of the samples we have was 10 samples. So let's say it was 1 second at 10 hertz.
Okay.
That's 10 seconds, 10 samples per second.
Mm-hmm.
So this is for one subject, for one infant. So what we do is, we take each of these, the feature vector at each of these time instance. So to be clear again, at each of these 10 time instance, we have a feature vector of some length. The feature vector is determined by the number of characteristics we decide to extract from the audio signals, including the fundamental frequencies and many other characteristics from the MFCC features. And so we take that feature vector of, say, some value, let's say it was 3. And we take those feature vectors, we decorrelate them, first of all, for training. And the goal really is to find in the high dimensional space, is there a space on which we can separate the instances from success samples from the time instances of failure samples. But when we do that training to try and separate them and find a good hyperplane, the high dimensional space, then on the evaluation point, we take, of course, a separate set of samples. So we don't use the same subjects we use in the training as the evaluation phase. In the evaluation phase, we take each of the instances, the 10 instances of a particular subject to be evaluated. We classify them based on the model that we are choosing to evaluate at this point. And ultimately, we then use another metric. So in our case, we explored both the use of just a simple majority count of the instances, but also we explored using another classifier, another support vector machine, to be able to classify the predictions. So it's like a meta classifier to be able to classify the predictions of each of the 10 instances. So each of the predictions become their own features to the next classifier. And then we use that to make one final prediction for that subject.
And is is the meta classifier method more performant than a majority or some kind of quorum-based system?
Yeah, in our case, it performs slightly better than just doing a majority count. And that's partly because it's able to take into account temporal dynamics now of the original instances in time.
Mm-hmm.
It's able to find the relationships between these instances over time, pretty much, and connect them with the ultimate outcome.
And the example that you use was 10 data points a second. Is 1 second? Is that was that just for illustration, or does 1 second worth of sample give you enough to identify this time varying indicator in the fundamental frequency of asphyxia?
Yeah. So one of the things that we've done is we use longer segments, but we first of all break them down into 1 second segments. So it's also a way of dealing with the fact that we don't have so many subjects, and we want to make sure that whatever we build is it leaves room for uncertainty. And so what we do is we break down the samples into 1 second segments, perform classification on each 1 second segment, and then average the results to give a prediction for that subject. So there's several levels of breaking down, including this. Yeah.
Okay.
So we at the very lowest level deal with the 1 second segments, and then we average that or ensemble that at the higher level to make a prediction for each patient. So in practice too, in the test we did with the mobile application, what we do is we record lengths of 10 to 20 seconds, and we break it down that way for the classification. So we split it. That also helps us to do parallel computations if the device supports it, and then we eventually combine the predictions. Yeah.
And in terms of where you are currently, what kind of results are you seeing with the method?
So the paper we have published, we obtained about precisely 86% of sensitivity. So that was the detection rate of infants who had asphyxia, and 89% of specificity. That was detection rate of infants who did not have asphyxia, who were healthy. And this is the one we published in 2015 that won the Best Paper Award at NIPS. At NIPS Machine Learning for Healthcare.
Right. And practically speaking, your baseline is against, in the field, it's against an unaided doctor trying to identify asphyxia just based on being presented with an infant that isn't responding normally.
Yeah. I mean, that's what would like to change, and that's not good enough to have the doctor just eyeball it.
Right.
But it should be clear that there's a gold standard. There's a gold standard which is used in Canada and in many other parts of the world.
And that's the blood oxygen level?
Exactly, the blood gas analysis.
Okay, right.
So they take the sample of the baby's blood usually through the cut umbilical cord, the arterial blood sample, and they analyze it for several parameters: bilirubin, acidosis, and electrolytes, and so on. And usually they combine this with the ABGA score of the infant. The ABGA score is a score assigned to literally every baby who has been born for the last 40 years. And it's a physical assessment of how well the baby is doing on 5 measures. So that's combined with this blood gas analysis to make a confirmatory diagnosis of asphyxia. And that
is the blood gas analysis also something that is routinely performed, or is it performed when there's a certain level of the score or some indication of a problem?
Well, in Montreal and the rest of Canada, it's a routine process for every baby that's born. And really, that's one of the things that makes it very convenient for us to do this study here. It's because we would not have to, as part of our study design, decide how we collect the gold standard. The gold standard is already a part of day-to-day clinical operations. We just have to read the charts to get that data. So yeah, it's routine here.
Interesting. And you mentioned that you're working on another project there as well, in addition to Ubenwa. What's that one?
It's called the APEX project. APEX as in A for APEX. It stands for Automated Prediction of Extubation Readiness. It has, I guess, one technical term, the extubation. And basically that refers to the process of weaning a child who is under respiratory support. Yeah, so usually infants who are born preterm, premature, which is usually around 7 weeks or thereabouts, are usually their lungs are usually not well developed or well formed to support spontaneous breathing for them, and so they usually require respiratory support. And this happens in general through the insertion of a tube down their trachea, and the other end of the tube is connected to a ventilator. And this ventilator provides oxygen to the infant at a certain interval, tunable by the physicians. But the physicians have to make a very critical choice under this setting. So they must decide when to remove the infant from this setup, from mechanical ventilation, as it's called, because the longer you leave the infant under that system, the increased chances of lung disease, because you've placed a plastic tube in the throat, in the trachea, and that causes there's interactions with the walls of the trachea and so on. And so that usually causes something called BPD, bronchopulmonary dysplasia, which effectively is lung disease. And that's bad because it's going to be a lifelong disability in most cases. So, but also you don't want to remove it too early. So that's the case when you leave it too long. If you remove it too early, the infant may not be ready to breathe on its own. And what could happen is that the infant would require re-intubation. So intubation is the process of putting the tube in, and extubation is the process of removing the tube. The infant could require re-intubation, and re-intubation is a technically difficult challenge for infants that have been intubated already, because there's swelling in the trachea and so on. And in some cases, they never succeed and the baby ends up having to die. And so there's this trade-off between when is the optimal time to extubate an infant under intubation in the ICU.
Can I ask the stupid question, which is, can't they remove the oxygen source without removing the tube and see if it works?
Well, that turns out to not be a stupid question.
Okay.
Because for our study, that's indeed what we are tracking.
Oh, okay.
Because one of the things the doctors do, as partly a way of evaluating how ready the baby is to disconnect the ventilator and let the child breathe through the tube first, to just observe how well the infant does. The challenge with this is that, while it's a good product, it's better than nothing, it still has resulted to currently in North America, the failure rate for reintubation is about 25%. So the doctors get it wrong about 25% of the time. And really, that's the core of the project. We're trying to fix that. We're trying to save those 25% of babies who end up either being reintubated or who end up with long-term disabilities.
So in a lot of ways, similar to the asphyxia problem, you have doctors that don't have an effective way of clinically assessing whether the baby is able to breathe on its own. And so they do, in fact, disconnect the ventilator without taking the tube out, but they're just eyeballing whether the baby looks to be ready. And in 25% of the cases, they get it wrong, take the tube out, and only then find out it has to go back in.
Exactly. Yeah. I say eyeballing, but, the doctors have many medical devices connected to the baby, ECG monitors, respiratory monitors, and so on. And, they look at this data, look at the patterns, and make an informed decision. And, that explains why they do still fairly well. 75% is not too bad, but it could be better. And that's really the goal of the project, to say, how can we take machine learning methods, apply it to an automated analysis of the heart rate signals, respiratory signals that we get from these infants during that spontaneous breathing trial period. So during that time when the ventilator is turned off, because that's usually the true measure of how well the infant might do when they don't have that ventilator on. So we take the signals at that point in time, and we are building primarily time series methods to analyze them and say, how can we make a prediction from this?
Okay.
Yeah.
And what stage are you in with this project?
We are at the stage where, as I mentioned earlier, in December 2015, we reached our target of 200 and I said 230.
Oh, right. Right.
Yeah. But that was the target of the project, anyways, to get to about to get to 200 babies for our training and development, and to get another 50 for validation. So those 50 will be left till the very end of the project. It's a 5-year-long project. We left till the very end to evaluate whatever methods that we've developed on the other parts. So we've done quite a bunch of analysis on the data over the last year, but really much of it has been much of the detailed work, I would say, has been starting this year, since we now have the minimal data sets that we require. Yeah, we've written about a few works we've done on the data in terms of using Markov chain models to model the respiratory patterns and how they change over time. we have a paper that we had published about this work last year at the Engineering in Medicine and Biology conference. Yeah.
Okay. And what was the conclusion of that paper?
Yeah, the paper was pretty interesting, or at least the finding we made from the paper was pretty interesting. So in that case, what we did was we did not look at the raw signals of how the heart rate is changing, the respiratory rate in particular. We looked more at states. So breathing in general goes through a number of states. There's 5 of them roughly. There's synchronous breathing, which is when the breathing in the rib cage and abdomen are in synchrony. It's very intuitive. So they're happening at the compression and expansion is happening at the same rate pretty much in both sides. There is a synchronous breathing when they're out of phase. That's the second pattern. The third pattern is the pause state, when the infant experiences a cessation of breathing at some point. And there is the movement artifact phase, which is somewhat a phase induced by external factors. Either the infant is moving or it's being handled by nurses and so on. And then there's the last phase is called an unknown state. And that is just the ones that don't fit into any of the 4 defined patterns. And so the goal of this project was to see how we could use switching models like the Markov chain, which models the transitions from one state to another, how we could take that and apply to this to learn more about what kind of transitions do infants who are ready for extubation go through, and what kind of transitions do infants who are not ready for extubation go through, and could this inform the classifiers we build in the future? And so it was more of a modeling task using chain models.
And do you think that the Markov chain model will play a big role in the approach you eventually take to create a diagnostic tool?
Yeah, indeed. On one hand, it emphasized some of the things that was known somewhat within the clinical community. That, for instance, pause states are bad. Cessation of breathing is not good because that means there's no power to drive the lung activity. So it reinforced some things like that. But also we observed some interesting trends in terms of the transitions from some of these phases to the others. That would eventually serve as features into the whatever classifiers we build in the near future.
Awesome. Well, you've been very gracious with your time, and we're running out of it. So I'd love to dig more into this, but I think in lieu of that, we'll just make sure that we have a link to this second paper here. But Charles, thank you so much for taking the time. You're doing some really interesting work. And I appreciated having the opportunity to learn about it.
Yeah, thank you very much, Sam. It was nice chatting with you as well.
Awesome. Thanks.
Cheers.
All right, everyone, that's our show for today. For more information on Charles or any of the topics covered in this episode, head on over to twimlai.com/talk/112. And remember to submit your thoughts on AI in your life at twimlai.com/myai. Thanks so much for listening, and catch you next time.